Good afternoon, everybody. I hope you're still awake before the next break. I'm talking about autoinflammatory interferonopathies and diagnosis and treatment. I'll go through a similar schedule as before. In this session, you'll be hearing about 3 autoinflammatory type 1 interferonopathies. I'll be talking about SAVI and CANDLE PRAAS. And Dr. Vanderveer, who's after me, will talk about Aicardi-Gouttiere syndrome. All these conditions are caused by mutations in genes that regulate type 1 interferon. CANDLE, or also called proteasome-associated autoinflammatory disease, PRAAS, or syndrome, is a condition that is genetically defined and caused by recessive or digenic loss-of-function mutations in proteasome genes. Let's actually see. At the moment, there are 5 genes known to cause SAVI. The proteasome has many more. So we are not able to yet genetically diagnose all the CANDLE patients, CANDLE-PRAAS patients that are out there. The prevalence of this condition so far is actually unknown, but less than 100 cases have been reported worldwide, and we believe that there is a degree that the condition is underdiagnosed. This condition was first described in the Japanese literature in 1930, and it was actually called first Nakayu and then later Nakayu-Nishimura syndrome. It's also called Japanese autoinflammatory syndrome with lipodystrophy, joint contracture, muscular atrophy, and so on. You can actually read this. I just put that in here because the older literature does not have a pathogenic term associated with it, and I left the references here as well. The clinical features of that condition that actually was also called chronic atypical neutrophilic dermatosis with lipodystrophy and elevated temperature, this is what CANDLE stands for, is a good description of the clinical features. Patients present usually within weeks of being born with fevers and nodular erythema. When these lesions are biopsied, they are panicolitis and neutrophilic and not vasculitic, although this actually could appear to be vasculitic, but it's a panicolitis. As they get older, the patches become more nodular and more defined, and you don't see these large lesions anymore. And as children progress, you start seeing the lipoatrophy, and lipoatrophy mainly in their face and also in their extremities. Patients have an interesting feature of a patchy myositis, which is different from JDM in its distribution. And they are at risk for the development of metabolic syndrome, particularly on steroids. And even low doses of steroids predispose. So while they lose fat in the periphery, you can actually see the thin rim of abdominal fat, which in somebody who has secondary metabolic syndrome due to obesity would actually be quite different, because you would see actually a large white area around here. And then you see accumulation of white fat here around the kidneys. So they accumulate fat into their abdomen. They actually can develop hepatic steatosis in many instances. And almost pathognomonic for all interferonopathies are the basal ganglion calcifications, which are more common in AGS, secondary than in CANDLE, and less common in SAVI, but can occur. And many patients have vascular calcifications. An underrepresented and insidious feature that you need to test for is pulmonary hypertension. And this is primary pulmonary hypertension, the absence of interstitial infiltrate or any previous lung disease. We are recognizing that more, and patients should actually be screened. As mentioned, genetic loss-of-function mutations in proteasome genes can cause CANDLE, and the genetic pattern is various. So it could be homozygous, 2 mutations or 2 identical mutations on the maternal and paternal allele, compound heterozygous, meaning 1 mutation on the maternal and another one in the same gene from the paternal, so 1 from father, 1 from mother, and digenic, meaning having a mutation in 1 gene that forms the proteasome and 1 mutation in another gene that forms the proteasome. So this is a very complicated way of inheriting the disease. The proteasome is a protein degradation unit that actually looks a little bit like a tube. And it actually is formed out of core molecules. Each one is encoded by a different gene. There are these beta proteins and the alpha proteins. And we have seen mutations in all of them, in the beta proteins and in the alpha proteins. And these are basically the components that can be affected. And all of those cause a decrease in function of the proteasome. The syndromes, depending on the gene involved, have now been numbered. So we actually go from PRAAS 1 to PRAAS 4. There are two assembly proteins that are important in putting those pieces together into that nice tubule. And there are three assembly proteins necessary. One is POMP, one is a heterodimer between PAC3 and PAC4, and one a heterodimer of PAC1 and PAC2. We have seen mutations in PAC2, which is actually encoded by GSMG2, and haploinsufficiency of POMP. That means you only need one loss of function mutation. It's usually a frameshift mutation that has been detected to get that, to get the disease as well. So basically, there is a fourth pattern of inheritance, and that is haploinsufficiency, which is inherited in an autosomal dominant fashion. So defects in these assembly molecules lead to defect assembly of the proteasome and the disease. We still don't know the exact mechanism, but proteins marked for degradation are literally fed into this tubule. There are proteases that actually chop the protein in small pieces and degrade it. And when that process is slowed down, we can measure these catalytic activity, chymotryptase activity and captase activity, and these mutations actually decrease the catalytic enzymatic activity of downgrading proteins. There is a threshold level which we don't exactly understand of proteasome dysfunction that triggers an interferon signature or interferon production that leads to the disease. We don't know exactly what the threshold is. We know that a heterozygous mutation present in a parent does not cause any disease, and we don't know the exact mechanism that actually triggers the transcription here of interferon beta and of interferon response genes, but not of IL-1 beta or IL-6. So there is still something to be understood, but dysfunction of the proteasome, regardless by which mechanism that reaches that critical level, causes the disease. And before I go into the pathogenesis, which is shared with SAVI, I'll tell you about SAVI, which is sting-associated vasculopathy with onset in infancy. SAVI is an autosomal dominant interferonopathy that presents early on with systemic inflammation, vasculitis, and vasculopathy, predominantly in cold-exposed acral areas, nose, also top the ears, and with progressive interstitial lung disease of variable severity. SAVI is caused by germline gain-of-function mutations, but there are now 2 patients described to get somatic mutations, somatic gain-of-function mutations that may not be detected by Sanger sequencing, but will be by next-generation sequencing. So if you have a strong suspicion and you don't find a mutation by Sanger sequencing, you need to do next-generation sequencing to make sure that there is not a somatic mutation that causes the disease. Again, less than 100 cases are described worldwide. The mortality of both of untreated CANDLE and SAVI is about 30% from published cases in the literature. These diseases have been not part, or actually have been a new addition and pointed to actually novel pathways or to type 1 interferon in causing innate immune dysregulation and phenotypes, and have really expanded our armatorium of drugs that we use to treat patients with autoinflammatory diseases. So CANDLE, again, presents with a vasculopathy or a vasculitis. You see actually blisters here, again, in cold-exposed areas. In some instances, it's actually called Chilblain's disease. Some patients have Raynaud's. And if untreated, this progresses to gangrene, and patients lose their digits and get scars in their nose or their ears. The vasculitis is very interesting in that you actually have a true vasculitis with neutrophils infiltrating and destroying the tissue, and there are also some immune complex, or it's a small vessel vasculitis. There are late in the disease immune complexes that I'm not showing you here. This is a typical lung picture that varies in severity, and you see the lung fibrosis. The lung disease can precede the onset of peripheral vasculitis, and we actually have 2 patients who have significant lung disease with actually known mutations who have hardly any peripheral vascular disease. And the opposite actually occurs as well. There's probably a genophenotype correlation that we haven't fully explored. These are the references that actually describe the disease, and then they'll be posted. So the mutation actually affects a protein called STING, or stimulator of interferon genes. It's a critical part in the viral sensing pathway that links the sensor cGAS, which actually senses double-stranded DNA viruses, to activation of STING through a mediator molecule called cGAMP that then leads to the activation of interferon. The mutations are in critical domains. They are in the dimerization domain. So this is one monomer, and this is another monomer, and they form like a flower-like shape. And the mutations are in that area and in an area 2, which recently has been defined. I'll show you that on the next slide. The mutations cause constitutive activation of interferon beta transcription, which cannot be further activated with something that activates the STING pathway, cGAMP, but you see that nice response to cGAMP in healthy controls. So patients actually are maximally activated without further responding to activation. Very recently, the molecular structure of STING has been characterized. It's actually 2 monomers, and you can see that green is from your side on the right side and yellow on the left. So when cGAMP binds, this part flips 180 degrees to the left-hand side, which then allows this molecule to oligomerize. Interestingly, our mutations actually make that neck supple, and without having to bind, without cGAMP binding being necessary, that 180-degree switch of that part of the molecule occurs spontaneously, and therefore the molecule aggregates and becomes activated in the absence of pCGAMP. I'm telling you that because many of the inhibitors, so STING has become a target for the development of drugs, and many of the drugs inhibit cGAMP binding. And you can now see that it would not work in these patients because they spontaneously activate STING in a cGAMP-independent fashion. So how do they present in terms of biomarkers? One of the novel biomarkers for autoinflammatory diseases is the interferon response gene signature. And you can see, you can measure it in multiple ways. This is by RNA-seq, looking at a larger panel of interferon response gene, and they are upregulated in CANDLE and SAVI, but not in NOMID and in healthy controls. In fact, it takes a long time to do RNA-seq and quantify that. So we've developed the nanostring score. There are many scores, yet you will find there is none that is better than the other. There are some reasons to use. Many of them are based on PCR and require considerable validation in different labs. Nanostring is a very simple and fast technique to measure transcripts in a sample, and they can be repeated equally here or in any other lab without having to do large normalization procedures. So it's very reliable, very reproducible, and it gives you a way to quantify the interferon score. We use 28 genes, and for a known interferonopathy to follow, you don't need 28. You actually only need 1 or 2 because they are highly concordant. However, our score has some that are not only activated by interferon but others, and they help us distinguish different diseases, and I won't get into that here. But there are some CANDLE mimics that are not just interferon-driven, where a ratio of these different genes actually is quite helpful. In contrast to the autoimmune interferonopathies, which include the complex diseases, lupus, JDM, and some vasculitis and scleroderma, there are no disease-specific autoantibodies that we find in the autoinflammatory interferonopathies. And while many of these conditions have immune complex deposition in the tissue that actually drives the pathogenesis and the tissue disease, and more activation of the adaptive immune cells, we mainly see the innate immune cells to be activated in, now I'm actually losing, I can't track this anymore. In the innate immune cells activated in CANDLE and SAVI, and it is mainly the monocytes in SAVI that produce large amounts of interferon beta. Up to recently, we had no treatments. Our conventional cytokine blocking treatments to TNF, IL-1, IL-6 have been ineffective, and we had no ways other than high doses of steroids to block interferon signaling in these patients until the development of the JAK inhibitors, which are approved for rheumatoid arthritis. They block multiple cytokine pathways, but the way they work by blocking JAKs is they basically inhibit, they bind, this is a JAK. The JAK has an ATP binding site, and these molecules sterically inhibit ATP from binding. Therefore, JAK cannot be phosphorylated, and it actually gets activated. The JAK inhibitors bind. Therefore, the phosphorylation is blocked. STAT cannot be phosphorylated. It cannot go into the nucleus and transcribe interferon response genes. And so we can use the absence or the down or the reduction of interferon response genes as a measure to see how well JAK actually blocks, JAK inhibitors block that pathway. So there are 3 currently on the market. None is approved for interferonopathies. Tofacitinib and baricitinib are approved for rheumatoid arthritis, and ruxolitinib for polycythemia vera and myelofibrosis. None is approved for the use in children, which has been a big problem. And we had conducted a clinical study in 18 patients, 10 had CANDLE, 4 had SAVI, starting with a very low dose, increase the dose to clinically optimal doses that were safe, and then continued on that optimized dose and analyzed patients. The study was done by Dr. Montalegre, and we published a mean duration of 3 years. The diary scores dropped, and the corticosteroid dose dropped, and in fact, let me just, I think maybe I removed that slide. And 50% of CANDLE patients went into complete remission. They were able to even 50% normalized their interferon scores. Now, this drug is very difficult to dose. It is not like a biologic where you can actually estimate the dose you give from the adult doses. But as you can see, we are giving higher doses in small pediatric patients than the adult recommended dose, which is 2 and 4 milligrams. The problem is the volume of distribution. Is very small. The kidney function is very good, and the excretion is fast. So the half-life of this drug is 3 to 4 hours for patients who are below 20 kilos, and it's actually 10 hours for an adult. So you can see that you have to dose more frequently in a patient, and as they get older, you can decrease the frequency. You also have to adjust for renal insufficiency. So they are very tricky they are much more tricky to diagnose, and they require more safety monitoring than biologics. But we've been able to actually get a treatment regimen. So this slide I thought came first, but this shows you that 50% of patients with CANDLE actually achieved complete remission, while we've seen improvement in SAVI, and in 2 other patients, none of them actually reached remission. So we need to do better. This is actually shown in different patient-measured outcomes that I won't go into. This is a picture in SAVI where you see improvement of the clinical findings. And what has been very encouraging is improvement in lung function, and the diffusion capacity actually improved, and the 6-minute walk tests improved. Pulmonary function tests in young kids are difficult to get, so we are actually, until they actually reach a certain age, so it's harder to actually get good PFT data, although we are seeing actually improvement. So this is very important because what is deadly in SAVI is the lung disease, although we would like to prevent that, and in fact we have seen until 2 months ago, we have seen no progression, and one patient actually started getting vasculitis in the cold and lost a little tiny bit of his fingertip. But overall, we see tissue protection with a JAK inhibitor, but we don't see normalization of the interferon score. Overall, the interferon scores do go down. This is mainly driven by CANDLE patients, and so is serum IP10, which is a downstream marker of interferon signaling. Safety is important, and why? So JAK inhibitors not only inhibit signaling through the interferon receptor, type 1, but they also inhibit interferon receptor type 2 signaling and a number of growth hormones, including erythropoietin and growth hormone. And I'll go fast because I've exceeded my time. So looking at safety, we see growth and height increase in patients on optimized treatment, and the bone mineral density increases, which has taken away some of the concerns about inhibiting growth hormone receptor function. Anemia can improve, and cytopenias improve in those that are cytopenic. It's important to assess BK virus. It's reactivated in the bladder, and there's no prophylaxis for it, but this is 5-year follow-up data, and the levels, if you dose at the levels we are and you monitor carefully, can be stable, but it is increased over baseline. In some patients who have herpes virus infections, we actually give prophylactic treatment. There are 14 SAVI patients who have received JAK inhibitors. All of them work the same and all see the same improvement in clinical features, but not normalization. In summary, autoinflammatory type 1 interferonopathies include the Mendelian disorder, SAVI, CANDLE, and Aicardi-Gouttiere. There are recent expansion of autoinflammatory diseases involving interferon type 1 signaling. They are characterized by certain clinical features that are listed here, and I had gone over. JAK inhibitors at the moment are first-line treatment, and so far the only ones that have shown significant efficacy in these diseases. The signature emerges as a tool that can be used to monitor and look at responses, and I mentioned the safety that is needed to look at. So thank you very much. This is the team, particularly the clinical team, who has done this work, and it would not have been possible without the collaborators who have critically evaluated these patients over time. The study had been going on since 2011. So thank you very much, and I thank the patients and also Lily for making this possible.